go. I'll start recording, and hopefully, can you guys see the question? Like big enough to actually read it at this point? Yeah, no? Yeah, you guys are good enough? Okay. All right, so I'm going to look at the 2015 question first. And this one tells us that blue food coloring can be oxidized by household bleach, which contains the hypochlorite ion, that guy right there. And then it makes products which it tells us are colorless. As represented by the equation above, the student used a spectrophotometer. One of the bad things about not being in class is you guys don't know what all of these are. Now, some of you guys may have used a spectrophotometer before in bio. I know there's a couple of labs you guys did in bio. Are you guys, well, you know what, do this really quickly in the chat. If you know what a spectrophotometer is, just say yes. If you have no clue, just say no. I'm just trying to get a sense of whether I really need to go over this a lot, because I know with the crazy stuff that's going on, there's some of you guys that have different experiences than others. Okay, a lot of you guys say no. Okay, so let me briefly explain what a spectrophotometer is, because it may be mentioned on the test. A spectrophotometer basically is a device that simply passes colored light through an aqueous sample, so a solution. Basically, certain molecules will absorb certain colors of light. So for instance, we're talking about blue food coloring. The reason that blue food coloring is blue is that this particular chemical that's here absorbs all of the other colors of light and reflects blue. That's why we see it as being blue. That's how we see any color, right? If a fire extinguisher is red, it's because it absorbs everything except for red, which it reflects. And so with a spectrophotometer, you would project this light through a solution that contains this material. And the more of this stuff that's there, the more blue that gets reflected, and the more of the other colors that gets absorbed. So then what happens is, on the opposite side, it's called a cuvette, is the small container. It's like a little square test tube kind of. It contains the sample, and on the other side from the light is a detector, and it picks up how much that light gets through. So what it basically does is it measures transmittance, how much light goes through, and absorbance, how much light gets absorbed. And the more of that stuff, the more of that blue material that's in that solution, if I project red light through it, it's going to get absorbed. The more of that stuff that's present, the less red light makes it through. Does that make sense? Because every time that red light encounters this material, which absorbs red, then the less of it is going to make it through, since that light was absorbed instead of transmitted. Now, I figured some of you guys may not have known what that is, so I did find a short video. Let me share this really quick with you guys. It's only like a minute or 2 minutes. I think it's about it. It's only 1 minute. So I'll just show this to you guys, and hopefully a picture's worth 1,000 words.
A lamp provides the source of light. The beam of light strikes the diffraction grating, which works like a prism and separates the light into its component wavelengths. The grating is rotated so that only a specific wavelength of light reaches the exit slit. Then the light interacts with the sample. From this point, the detector measures the transmittance and absorbance of the sample. Transmittance refers to the amount of light that passes completely through the sample and strikes the detector. Absorbance is a measurement of light that is absorbed by the sample. The detector senses the light being transmitted through the sample and converts this information into a digital display.
Okay, so I mean, it's pretty simple, really, when you get down to it. You're shining light through a sample, some of it's absorbed, some of it's transmitted. That's really it, okay? So it's simply a way of measuring how much of a particular compound is in a sample. So anyways, that's how it worked. It's set to a wavelength of 635 nanometers. And this is, again, a wavelength. I don't remember, if they don't require us to do, well, by the time we would have went over this in physics, we were already in distance learning. So I don't know, I don't think we went over that last year, but I usually do. But just so you guys know, when we talk about wavelengths, and darn it, I need to share screen again. Basically, when you take the whole electromagnetic spectrum, it goes from really long waves, which like 100 meters plus, so more than a football field. Those are radio waves. When you get into the waves that we use for cell phones and radar and those kinds of things, they get shorter and shorter and shorter. Infrared, which we sense as heat, is going to get down to 1/100th of a centimeter, which we're getting really small now. Nanometers are even smaller than that. And here is our entire lens on the world. Basically, everything that we see, you guys, is between about 375, 380 nanometers. That's going to be things that look violet, okay? All the way up to almost 750 nanometers. And notice the different colors have different wavelengths. And so when this tells us that we are at 635 nanometers on this question, we're right about here. So we're pretty much orange light is what we're projecting through that sample. Now, why would that be appropriate? Because if you think about orange, if it hits something that looks blue, that substance is going to absorb orange. Does that make sense? Remember that the way that we see colors, we don't see what's absorbed. We see what's reflected. And so what I would say is that blue food coloring is going to reflect blue, but absorb orange. So the whole idea here is the more blue food coloring that I have, the more of that orange light that I'm shooting through that sample is going to be absorbed. Okay? If I don't have much blue, that orange just goes through pretty much, like if you take distilled water, which is colorless, and you project orange light through it, whatever amount of light you send through it is going to end up hitting that detector on the far side. And so if I put something that absorbs orange in there, I'm going to get less and less and less orange light making it through to the other side. That's basically how those things work. Okay, enough of that. I've explained how it's set up and haven't really answered the question yet. So this tells us it's set at a wavelength at 635 nanometers to study the absorbance of the food coloring over time during the bleaching process. So what I would expect, and the reason why they're going to do this with a spectrophotometer is they can use the readings, the absorbance, to tell how much of that blue stuff, that blue food coloring, is left. Because as it goes to colorless, it's going to stop absorbing the orange light. Okay? I hope that makes sense to you guys. It's a fairly simple process, really. So in the study, bleach is present in large excess so that the concentration of OCl, the hypochlorite ion, is essentially constant through the reaction. So what they're basically saying is the amount of hypochlorite that you have is going to be so high that the amount you lose due to this reaction is negligible compared to what you started with. So it won't slow down because the reaction won't slow down because you lose so much OCl that it becomes limiting. The limiting reactant is the blue food coloring. the student used data for the study to generate the graphs below. Now, these should look familiar from the last chapter that we were working on. Okay. Basically, anytime I see a graph on a question like this, you guys, I'm looking at the axes. And what I see here are some fairly familiar terms. What they're basically, although this is not a graph of concentration, this is a proxy, a stand-in for concentration, because the absorbance is going to increase as the concentration of the blue dye increases. Does that make sense? It's going to absorb more of the light as you have more and more blue food coloring molecules there. So as I lose the blue food coloring, as it reacts with the hypochlorite, you're going to get less and less, which means I'm going to get less absorbance and more transmittance. So you can see that kind of makes sense given what you're seeing here, that over time, I started with a high level of absorbance and it dropped exponentially, essentially. Okay, it dropped fairly quickly because I don't have as many of those molecules later on. But so I can think of this pretty much like the concentration of that blue dye. This is the ln of the concentration of that blue dye. And this is 1 over the concentration of that blue dye. Now remember, what we're looking for on a question like this is we're looking to see which one of these three graphs is going to give us a straight line. And clearly, it's this one here. Now remember, if the concentration versus time, if that was a straight line, this would have been 0 order. Okay, if it was a straight line. If the ln of the concentration, which again, we're just looking at the absorbance as a stand-in for concentration, this would have been first order. And if this was a straight line, this would be second order. Okay, so clearly because this gave us the straight line, that this particular reaction with respect to the blue food coloring is first order. How many of you guys got that? Did you get that this was first order? Okay. I mean, really, as soon as you see what's on these axes, you guys, that's going to be a dead giveaway. And the thing that I noticed between these two questions is essentially that one from the 2015 test, it's the exact same part A. You're interpreting graphs is what you're doing on both of these. So if you just remember which one of these goes with first, second, and third, and be careful, or excuse me, 0 first and second, be careful because sometimes they shuffle them around. They didn't on this one. They put them in order pretty much. But sometimes they'll take this and put it here, and they'll take the ln and put it over here. So pay attention to which one's which, okay? All right, so anyways, that was first order on that first question. And I will tell you guys that if I look at their guidance for scoring, that's worth 1 out of 4 points. There are a total of 4 points on this question. It's what they call a short question. Their long questions tend to be 8 or 9 points. So on this one, if there's 4 points total, I need to get probably 2 of them to look at it and say, hey, I would probably pass if I could do this on every question. The second one says, or part B says, the reaction is known to be first order with respect to bleach. In a second experiment, the student prepares solutions of food coloring and bleach with concentrations that differ. What they're trying to do here, more than likely, if you think about this, if they're trying to make concentrations that differ from the first experiment, they're probably trying to do the method of initial rates to actually figure out what K is. That it doesn't ask you about that, but that's probably why you would do that. When the solutions are combined, the student observes that the reaction mixture reaches an absorbance near 0 too fast. in order to correct the problem, the student proposes the following 3 possible modifications to the experiment. So the problem is the reaction is occurring too fast. All right. If you were to say that this was answer A, this was B, and this is C, which one did you guys pick? Just in the chat, throw it in there really fast and tell me which one of those 3 that you would select. So far, B is the clear leader. The correct answer here is B. So what this part was about, you guys, and before I get too much further, do read all your directions. So for instance, if I look at A, it said based on the graphs, what's the order? It didn't ask you to explain. It just asked what the order is. Don't give them any more information than they really ask for, is what I would say. Because the more BS you try to throw at them, the more likely it is you're going to put something in there that's wrong. So if all they ask you to do is to identify something, just identify it. Don't explain it. If they say justify your answer, then you got to explain why. Okay. In this one,
So Mr. Gibbs, sorry, does it have to be complete sentences, or like you said, just the answer?
Yeah, if you do not have to write complete sentences here. Like for instance, on A, I don't have to sit there and write out a whole sentence on why I said this. If they ask you for that, just give it to them. You're going to have enough work to do, and you're going to be a little bit stressed for time, which that's natural on these exams. But just answer it and move on. Okay? These are chemistry teachers that grade these. They're not English teachers. They don't care. Fact is, they probably really don't like having to write complete sentences. That's how chem people are. So yeah, you can just answer. I would say do exactly what it is that they're asking for, and don't worry about doing a whole lot more. So anyways, this one was B. Remember, the problem is that the reaction was happening too fast. So I'm looking at these and simply asking myself, which one's going to slow this down? And A is clearly not the answer, because almost every time you increase temperature, it makes the reaction go faster. So there are very few reactions where increasing temperature actually would slow it down. Okay? Increasing the concentration of the bleach, what this would do is, if you think a collision model, this means the bleach is going to collide with the blue food coloring more frequently. Does that make sense? That if I increase the concentration, if there's more bleach in there and I don't change anything else, then I'm going to see a situation where I have more bleach, which means more collisions between the bleach and the blue dye, and that means an even faster reaction. So this one's faster also. So if they really ask me, you do have to explain on this one. But I know right off the bat that A and C aren't my answer. Both of these make it go faster. How is increasing the concentration of the food coloring going to slow this down? Well, what I would say is something along the lines that more particles, you know what, I already wrote this down, so I'm just going to show you what I wrote. Here's what I wrote for my explanation. So I would circle increasing the concentration of the food coloring. Didn't close my quotations. The additional, and then my explanation would be the additional food coloring would take longer for the bleach to react with, since additional collisions would need to occur to completely remove the blue dye. Does that make sense? If I have more blue dye particles and the same number of hypochlorite molecules, I'm going to have more collisions in order for all that blue to leave, to be gone. Something like that. That's all you're really going to need. This question, or this portion of the question, you guys, was worth 2 points. If all you did was get B right, you would get a 50% on this question, and that would be passing. That would probably get you approximately, it's a high 3 or a 4 is the typical percentage. So I'll tell you exactly what they said. If the initial concentration of the blue food coloring is increased, more time is required regardless of the reaction order for the bleach to oxidize the additional blue food coloring. They were a little more eloquent than me, but it essentially says the same thing. Okay. And again, you would get 1 point on this question for giving the right answer. So if all you did was answer B, but you totally guessed and had no clue about why, you would at least get 1 out of the 2 points. Okay. I will tell you that a lot of people that don't pass the test, well, pretty much if they don't review on their own, they don't pass. It's I mean, that's up to you guys. That's your guys' choice, what you guys do with that. But you got to do some review on your own and figure out where your own personal weaknesses are and get them fixed, is what I would say, if you really want to have a chance to pass. The other thing, though, that I see is that I have some people that basically didn't prepare themselves, and so when the test starts, they just put their head down. Or they run into the first question that challenges them, and they just throw up their hands and don't press on. So there were other things they could have answered, but they basically made a choice not to be successful, is what they did. And that's your choice, again. But again, if I didn't know how to do A, but I had a clue on B, I'd be kind of pissed at myself if I skipped over all of it and moved on and didn't answer, because this part could have meant the difference between passing and not passing. Okay. I will tell you, I don't think he'd mind if I said it, but my nephew, Joe, who graduated last year, he studied. I helped him on a few things, but a lot of it was him kind of getting us a prep guide and going through it and figuring out where he was weak. I think he got a 4 on it. He's at University of Washington right now, and he skipped 2 quarters of chemistry. He has to take 1 quarter, which he's taking right now, and he'll be done with chemistry for his entire major. So there is a reason why you would want to pass it, but whether or not it's kind of you do it is up to you guys. Okay, let's move on. I'm going to waste all my time doing this. On to C. In another experiment, kind of put this out of the way. In another experiment, a student wishes to study the oxidation of red food coloring with bleach. How would the student need to modify the original experimental procedure to determine the order of the reaction with respect to red food coloring? Well, here was the procedure up here. And I will tell you that, remember we mentioned what this wavelength was and the fact that the light that you pass through it has to be absorbed by the substance that's in there. So if I think about red food coloring, that means it's reflecting red and it absorbs everything else. Now, I wouldn't be all that specific, but what I would say here, I would say something like, I would need to adjust the spectrophotometer wavelength. You could also say frequency here, you guys. They're related to each other. So I would need to adjust the spectrophotometer wavelength, and I wouldn't specifically give a wavelength. I don't think, I haven't looked at the guidance on this one yet, but I don't think they're going to make you specifically say, hey, I'm going to adjust it to 500 nanometers or anything like that. I would simply say, I would need to adjust the spectrophotometer wavelength to a value that is appropriate for red light. So again, I'm saying how I would modify it, but I'm not saying the exact value. I think this is going to be okay. Maybe I'll be wrong. Let's see what they said in the guidance. Yeah, actually, it's pretty basic. The spectrophotometer should be set to a different wavelength. That's it. That's worth a point. All right, now, let's say I had no flipping clue what a spectrophotometer was or how it might be used or how it works. Let's say I had no clue. What's a clue that maybe, and I'm telling you guys, they're guessing strategies. The fact that I'm given a numerical value here means there are other numerical values that could be. So if you really think about the way that they set this up, anytime they give me a numerical value like this, whether it's a temperature, whether in this case it's a wavelength of light, that's something that more than likely can be adjusted. So if you're to the point where you're just throwing a guess at something, then that would be a likely suspect for something that I would throw a guess at. Okay. So that's this one. That's this one, 4 points. I think the first one, based upon what we've done in this chapter, this one was pretty easy. As if you looked at what these axes were telling you, immediately if I would see these and I see two curves in a straight line, I'm thinking about how we, with integrated rate law questions, how we have to use the graph to help us to determine what the order is, what the reaction order is. And so my immediate thought is I see one that's a straight line and the other two that are curves. I'm going to focus in on this one. I'm going to look at the axes and this is going to tell me which one's which. So there's a little bit of information you have to know and then you use that to interpret what you got. Anybody get all three of them? All three of the questions right? Or maybe I should talk about points. Anyone get all 4 points or feel like they would have? You can throw it in the chat if you don't want to brag, but I will tell you that if it was me, I'd be bragging to everybody because that's how I am. I'm impossible. So a few of you guys did. Okay, anyone get 3 of the 4 points? 3 of the 4? Yeah, a couple of you guys got 3 of the 4. Good. Guys, if you can manage, like I said, to get 2 out of the 4 points on every question, or half the points, you're going to pass this test, is what I would say. How about 2 out of 4? 2 out of 4 should get you. Okay, good. So I would say, based upon numbers you guys gave me, roughly half of you guys got 2 out of 4 or better. That's good. That's awesome. Okay, let's move on. Now, hopefully, this second question will be a bit easier. We'll see now that you've seen the first one. You know what? I have a timer that's going off. I need to shut it off because it's driving me nuts. Alexa, stop. Okay, so nitrogen dioxide, NO2 is produced by byproduct of combustion of fossil fuels in internal combustion engines. We've seen this before when we talked about photochemical smog. This was one of the things we saw when we talked about photochemical smog back in chapter 5, I think it was, on gases. Anyways, at an elevated temperature, NO2 decomposes according to the equation below, and there it is. The concentration of a sample of NO2 is monitored as it decomposes and recorded on the graph directly below. The 2 graphs that follow it are derived from the original data. So what did I say about graphs? I said, look at the labels on the axes first and foremost. This is giving me concentration versus time, all right? This is 1 over concentration versus time. And this is the ln of concentration versus time, okay? Now, immediately, again, when I see these, I'm thinking that this is going to be a reaction rate question because these are the graphs we look at for reaction rate questions. Now, if this one gave me a straight line, all right, if this graph gave me a straight line, what order would this reactant be? If this first one gave me a straight line, what order would this be? Yeah, this is going to be 0 order. Okay, that would be 0 order when it's concentration versus time if it was a straight line. But I can see that if I actually graph these points, I really like on these questions that they don't make you actually determine these values, but they give them to you. Not that you couldn't determine 1 over the concentration, but it's time consuming, and that's something that you don't have. So the fact is, all you're really doing here is connecting the dots on these. Means that I'm really just worrying about interpreting the graphs. I'm not having to do the graphs. So what about this one, guys? If this is a straight line, which it is, what's our reaction order if this is the graph that gives us a straight line? Good, this is second. Several of you guys answered. Now notice they didn't put them in order here. So you really want to know which of these go with which reaction order. And this one, the ln would be first order. Okay, so in our case, and for letter A, they ask us, explain how the graphs indicate that the reaction is second order. Now they even tell us it's second order, but I would simply, all you really have to say on here is that a graph of 1 over concentration of NO2 versus time gives a straight line. All right, pretty sure that's all you're going to have to say on this. Let me find the guidance. Yeah. Here's what they said, you guys. Linear graph of 1 over concentration NO2 versus time indicates a second order reaction. That's pretty much what I said. Again, just a little more eloquent than me. Write the rate law for the decomposition of NO2. Well, if we know that this is second order, okay, and I know there's only one reactant here. It's just the NO2. That's essentially it. Now, there are 2 particles of it. I'm just going to write the generic form of a rate law for a second order reactant, which would be rate equals K times the concentration of your reactant, which is NO2, to the second power. That's it. Okay. Now, the first question, where did it go? That first question here, which was just interpreting the graph, this was worth 1 point. This was another 4-point question. Writing the rate law, that's worth another point. Okay. So if you were able to tell this was second order, that graph was second order and explain why, and you could write a second order rate law, okay, just like this, you're at 2 out of 4 and you're going to pass this question because it's only worth 4 points total. Okay. Now we're going to go to this one. And this one deals with reaction mechanisms. So this one asks us to consider 2 possible mechanisms for the decomposition reaction. Is the rate law described by mechanism 1 shown below consistent with the rate law you wrote in part B? Okay, now remember that when we're looking at whether or not a mechanism is appropriate, it has to do two things. Number one, it has to agree with the overall reaction. So what I'm going to do first off, I'm just going to kind of draw a line right here. And I'm going to eliminate species that are on the opposite side. So I can see right away, and I'm just going to kind of shortcut this one, that this and this, they're going to end up canceling. NO3 is an intermediate, okay? You remember we went over the definition of an intermediate. That's essentially something that's created in one reaction and then consumed in another one within the mechanism, so it doesn't show up in the overall reaction. And then I'm just going to write what's left over. And I've got two NO2s here. And it's going to yield two NOs and one O2. So my first test to see if this is consistent is, how does that compare with the overall reaction? Well, it's exactly the same thing. So the first test is passed, I would say. Do you guys remember the second test? What you have to do now is I have to take the rate determining step. So I would say, does, I'm going to call it RDS, rate determining step, match the experimentally, I'll just say match the experimental rate law. And we have an experimental rate law already. That's the one that we wrote right here. Okay, so using molecularity is what you want to do here. If I look at this reaction, the slow step, the rate determining step, what's the molecularity here? All right, is it unimolecular, bimolecular, termolecular? Well, anytime I have to have two collisions between two separate molecules, this is going to be bimolecular. Okay, this is bimolecular. And the rate law for a bimolecular step would be rate equals K times the concentration of that reactant. In this case, it's the same reactant, to the second power, because I have to have a collision between two different molecules. So what would you guys say? Does the rate law that I would get from the rate determining step, which is this right here, does it match what we got from the experimental value? The answer is yes. And so for this one, it meets both requirements. And so what I would say is yes, that it is consistent. Okay, is consistent, and I would say because mechanism gives overall reaction. I'm just kind of shortening this, you guys. So when I say it gives the overall reaction, I'm saying that we added these two together and we got the reaction that we're looking for. Okay. And the rate law from the rate determining step agrees with the experimental rate law. Sorry, I know it's a mess, but it's there. Okay. I will tell you what they wrote. I'm sure theirs, again, is more eloquent than mine. It says, yes, step one is, well, I'll let you guys can read. Kind of uncover mine too. Yes, step one is slow. Therefore, it is the rate determining step of this mechanism. The rate law of this elementary reaction is rate equals, and then it basically spells out what we wrote. And you're essentially using that molecularity, which is consistent with the second order rate law from part B, which is the experimentally derived. Okay. So this one is worth 1 point. One, yeah. Seems like a lot to do for 1 point, but it's worth one. So if you would have gotten that one, if you would have remembered how we test these different mechanisms, you would have got, some of you guys would have got 3 out of 4, at least at this point. This one, I kind of hesitate to go over it with you guys only because it uses equilibrium, which we haven't gone over yet. So this part of the question is really something that you have to have done the next chapter in order to really finish up. But I'll briefly talk about it. Okay, so what did you guys think of this one? Was this one easier or more difficult than the 2015, the first one that we did? Yeah, easier? Maybe part of it is that we just went over the other one, and so the stuff's a little more current in your brain. But I didn't personally think that either one of these two were all that bad. So let me ask you guys again. And instead of saying it's 4, let's just forget about this one for right now. Of the 3 points for the first 3 parts to this, anybody get 3 out of 3? Anybody get 3 out of 3? Brag a little bit to me. Nice, nice. Good, good, good. All right. How many of you guys got 2 out of 3? 2 out of 3 on this. Of course, 2 out of 3 would definitely be a passing score. Good, good. Awesome, you guys. All right. So it looked to me like it was maybe about 8 or 10 of you guys. So a little less than half on this one. This one here is kind of complicated, and I really kind of shudder whether I want to explain it or because I'm afraid it's going to confuse you. The issue that you have here is that your rate determining step is this one. And clearly, if I include the N2O4 in my rate law, like we did here, if I include the N2O4, it would be unimolecular. And so I would typically write a rate law, which would be rate equals K, concentration of N2O4. Now, here's the issue. This totally does not match, and it would be first order because this is unimolecular. It totally doesn't match what we got on the bottom of the previous page. At least it looks that way. But if you think of it in a different way, look at the fast step that occurs just above this. Well, and this reaction goes back and forth. It's an equilibrium situation that is achieved very quickly. It says fast equilibrium, which essentially means that you could say that one of these is equal to 2 of those. Because again, this reaction is going to go this way, but it's also going to go back the other way. And when it does, it's going to give me 2 of these. So we could essentially say that these are equivalent to each other. And that would mean that instead, if the NO2s is equal to 1 N2O4, then I could basically substitute these 2 in for reactants in this reaction. But that requires you to know how to do equilibrium statements, and we're not there yet. So we're going to learn how to do that in the next chapter. I don't want you to get too stuck on this one. But I could easily see where you would look at this and say, well, this doesn't match the other one, so it can't be a possible mechanism. Okay, so without knowing that, it's kind of hard to do that last one. I do want to leave you guys with this on these questions, and that is that remember that just because a mechanism is possible, just because it meets these two requirements, does that necessarily mean that it absolutely positively is the mechanism for the reaction that they're describing in this question? Is it 100% positive that that's the mechanism? If it meets these two tests? The answer to that question is no. We can't know for sure whether this reaction, these steps, this mechanism is the one that's actually occurring. All we can say is that it's possible. We'd have to do some other tests. Like for instance, if I have the ability to measure the concentration of NO3 during the reaction. Now notice what would happen to this concentration is when I first began the reaction, there'd be none of it present because it's a product in this first reaction. So when I first start, none of it would be present. But then let's say that we have the ability to use a spectrophotometer and monitor those values as the reaction progressed. Well, I would see this stuff would start to get made. So I'd see a little bit of a spike in that material as the reaction progressed. But then because of the second reaction, it would go away. So if I could possibly see the presence of an intermediate as the reaction occurred, and that intermediate matched this intermediate that we see in our mechanism, that's another piece of evidence that would say, yeah, this may be the right one. But I won't ever be able to know for sure, you guys. We're dealing with levels of confidence is what we're doing, okay? All right, I'm gonna stop because we're pretty much just out of time, and this took a lot longer, but hopefully you guys got something out of this in terms of picking up some extra tips and tricks and learning how a spectrophotometer worked and that kind of stuff. Okay, so I do have an assignment, and I'll make this due tomorrow, so it'll be your off day assignment, I guess I'll say. I'm gonna go ahead and share screen. Let's go back over here. Get you guys. Oops, that's last period. Don't look, don't look. Not that you guys care, it's someone else's scores, not yours. If I go over to modules, we're gonna move on to a new chapter. Again, doing a big fat test at the end of each section takes a lot of time, and I'm feeling the crunch now as we're starting getting towards March. And I want to make sure that we get through as much as we can. So instead of spending 2 days on a review and a test, we're going to move on to the next section. You guys are going to be doing a crossword for chapter 13, just to kind of get you used to the stuff that's in there and where it is. It's not a huge one. It's only got 13 total words, which that means something, should mean something to you specifically. It means this chapter has a lot of stuff to do and not that much stuff to know, if you get my gist here. Okay, this isn't going to be loaded with a whole bunch of little trivial details. It's mainly going to be doing stuff. Okay, and you'll see equilibrium is not that hard, which is what this chapter is about. So I want you guys to do this. Let me go back to modules, because I think I was actually going to have it due today, but because we spent all the time on the questions, I think I'll change it to tomorrow. So edit. Let's call this down to due date. Let's call this the 12th. And I'll leave it at 9 p.m. I'll give you guys all day to do it, since I won't see you anyway. But do notice it's going to close at midnight. So make sure you guys get it done tomorrow for points, because after midnight tomorrow, it won't let you turn it in. So make sure you guys get that done. You might work on it today if you get some time. But that's about it, you guys. So I'm going to end it right there. If I go back to modules, do notice, though, you can take a picture of those two FRQs that we just did. Okay, these two PDFs. If you take a picture of those two and submit it, and you can do it right on this assignment from yesterday, you can submit pictures of those and you'll get 5 points. Really, it's mainly a participation score in that you took the time to write it down and consider the question. Okay, so you do want to do that today and do it by 1 p.m. So do it right now, get it done and over with. Let me stop the share and bid you guys a fond farewell. So you guys have a great Friday and a great weekend. And yes, there was a question, can you leave the last part of the question blank so that letter double I never know how to call out those things. So part C, letter double I on the 2019 FRQ, you can leave that blank. It won't take points away from you. Okay. All right, I'm going to shut up so you guys can get on your way. Enjoy the rest of your day, you guys. Have a great weekend, and I'll see you guys on Tuesday. Take care.
Mr. Gibbs, I have
Yes.
a quick question. Is it okay if I typed it? Because I typed it yesterday on a doc. Is that fine? Okay, I just wanted to double check. Okay, great. Thank you.
Easier to read for me.
Okay, great. Thank you. Bye, Mr.
See
Gibbs.
ya.
Have a good day.
You too.